Hello, guys. We're going to talk about genetic mutations today and how they occur. Generally, a DNA sequence is altered during the mutation. A nucleotide is changed with another nucleotide, and resulting mRNA is altered, and a different protein is produced which is not desired. So, two types of mutations we can talk about. One is called germline mutations. They occur in germ cells and they are passed on to the offspring. Somatic mutation, they occur in normal cells and not passed to the offspring. And mutation can be caused by radiations, chemicals, and viruses. So let's talk about type of mutations. We have point mutations. When a single pair nucleotide substitution occur with another nucleotide, non-desirable nucleotide. So there are three type of point mutation, missense mutation. So as you see, this is the DNA structure for a codon, and right now it's producing leucine protein when it goes through the transformation and translation. So when this particular nucleotide, which is thiamine, is replaced by guanine, the resulting mRNA is changed, and also resulting protein is changed. So this is undesirable effects. So this is called missense mutation. Nonsense mutation is basically when replacement of a nucleotide, which occurs right here, produces stop codon. So stop codon is the codon that stops the synthesis of protein. Initially, it was producing cysteine, that's the name of the protein, but this mutation caused this particular codon to stop the synthesis. So this is called nonsense mutation. Silent mutation is when a protein is produced, and when that mutation occur right here, the resulting protein does not change. It's the same protein. There are two codons that produce the same protein. So that's just a silent mutation. Now we're going to talk about frameshift mutation. Frameshift is when, so this is a series of codon, a series of nucleotides, right? So you can, I'm splitting into three each, so I can give them names. Basically, it's a series. There's no gap in between here. So when this insertion occurs, so this insertion is occurring right here, that shifts everything to the right. So now, since this insertion, that insertion comes right over here. Now this becomes CGA, and this is arginine, and before it was glycine, and this was arginine before, and this becomes alanine, and this was met, and then it became a different protein, different amino acid. I'm sorry, different amino acid, and this is left over right here because of the push to the right. So this is very serious mutation. It's called frame shift. It shifts the frame to the right because of the insertion. Now we call another one is called inversion. Inversion is when this sequence right here is kind of turned around, kind of in this fashion. So A comes here and C comes here. So it becomes AGC, which is serine. Before it was arginine. It was arginine, now it becomes serine. So now the whole series of codon are produced are different for the final protein that is going to be produced. So that's inversion. Translocation is occur a set of chromosomes going to exchange their genes like this, in this format. This particular section goes over here, and this particular section comes over here. And the resulting chromosome, which was longer, chromosome 6, it was longer, now it's shorter. And chromosome 22, which was longer, which was shorter, now it becomes longer. So it's called translocation. So these are the type of mutation that can occur in your chromosomes or in the DNA. Let's recap quickly that what are genetic mutations are. There are two types, germline and somatic. And the types of mutations are point mutation that produce three types of point mutations. Missense, when different protein is produced than the desired one. Nonsense, when a stop codon will stop the processing of proteins. Initially it was cysteine supposed to be coming out, but now it's a stop codon is stopping the synthesis of protein. Silent mutation, end result is same because of that mutation. Frameshift is a serious one. It shifts the frame to the right because of the insertion occurs. Inversion, the codon shift their position, and the nucleotide, as a result, nucleotide shift its position. Translocation, two chromosomes share their, or you can say exchange their part of their chromosome structure with each other and cause their mutation. Hopefully you like this tutorial of genetic mutations. If you like, go ahead, subscribe the channel and like it or comment it. We'll see you in the next tutorial. Till then, have a good day.